Hi, my name's Dr. Christopher James Ward from the Kansas University Medical Center. And today I'm going to talk about the development of a calibrated real-time reverse transcriptase polymerase chain reaction, an RT-PCR, for Cystogene mRNA. Now, the most important word in that title is calibrated. We wish to determine the number of message molecules in a sample of RNA, and then from that, an approximation of how many copies of that mRNA we have per cell from a given tissue or cell line. Now, we're particularly interested in the genes for the human polycystic kidney diseases. And these can be split into two groups. The autosomal dominant forms, which are responsible for disease in about 1 in 800 individuals. These people develop cysts in the latter part of their lives and will require renal replacement therapy. In the case of Pkhd1, this is in the 5th decade, and in the case of Pkhd2 mutations, this is in the 7th decade. Now, there's also a severe autosomal recessive form of polycystic kidney disease that presents at birth and causes cysts in the kidneys and fibrosis of the liver. And it's due to mutations in the polycystic kidney and hepatic disease gene 1, Pkhd1. Now, I'm also interested in an orphan ORF open reading frame called C21orf62, which encodes a protein called exosomal polycystin 1 interacting protein. And as the name suggests, this protein interacts with polycystin 1, the product of the Pkhd1 gene. Now, what we want to do is we want to develop an assay that can count the number of message molecules we have in a particular sample. Now, to do that, we need to calibrate our assay. And to make a calibrant, we have to actually make a synthetic RNA that mimics the messenger RNA that we'll find in the animal or the human being. And so what we do is we make a piece of RNA that's about 500 base pairs long that is base perfect when compared with a segment of the naturally occurring mRNA. Okay, and so here we have this synthetic RNA here. Now, we're going to make a real-time assay for 4 different genes: Pkhd1, Pkhd2, Pkhd1, and C21orf62. So we'll have to make RNAs for each of these messages. So once we've made the RNA, we can then purify it, work out how much we've got, and because we know the molecular mass of the RNA, we can work out how many moles we've got, and then using Avogadro's number, determine how many copies we've got in a microliter. And usually we end up with about 10 to the 11 copies per microliter once we've finished this protocol. Now, the idea is to put this in a tube and then make DNA from it using an internal primer and reverse transcriptase to make this red DNA strand. And then what we're going to do is we're going to do a TaqMan assay. So this is the guts of a TaqMan assay here. You have a forward primer and a reverse primer and a TaqMan. Now, the TaqMan is interesting because it's a piece of DNA, and at one end it has a quencher, and the other end it's got a fluorophore. Now, these are so close together they interfere with one another. So the quencher is within the FRET distance, the fluorescent resonant energy transfer distance, and so it suppresses the fluorescence from the fluorophore when in solution. When this binds to DNA, it can become the substrate for the polymerase going in a forward direction here. The exonuclease activity of the polymerase will degrade this linker, and this fluorophore will fly off and become fluorescent, and the quencher will be diluted out. And so with every round of PCR, you'll double the amount of fluorophore that you've got. So the idea behind the assay is that if you start off with a small amount of RNA, it will take a long time for you to accumulate free fluorescent dye. So it will take multiple cycles. But if you start off with a lot of RNA, it'll only take a few cycles. So let me explain. In the real-time PCR machine, we have a fluorescent threshold, okay, which can be thought of as a finishing line in a race. All right. So what we've done here is we've taken a piece of RNA, like this one here, from the Pkhd1 gene and set up a TaqMan assay for it. And here you have 10 to the 9 copies in a tube. And as you can see, this has crossed the fluorescent threshold, the CT, at about 9. Okay, and then this is 10 to the 8, 10 to the 7, 10 to the 6, 10 to the 5, 10 to the 4, 10 to the 3, 10 to the 2, and then about 10 copies here. And what you can see is that with every jump in tenfold dilution, the CT increases about 3.32 fold. So if you were to have a blank that was crossing at 18, you would know that you had roughly 10 to the 6 molecules there. Okay, but if you crossed at say 30, you would only have around about 100 molecules in your tube. And so using this threshold, the CT threshold, we can determine how much message we had in the original tube. Now we're not just going to do it with one color on one gene. We're going to do it with 4 different colors, and this is what you get. We're looking at EPSIP, which is that C21orf62 gene, Pkhd1, Pkhd2, and Pkhd1. And here you can see they're all kind of overlapping, and they're all reaching a threshold roughly together. So this is really what you want. So in one tube, you can actually measure 4 different messages. So how do we set this up? Well, like all things in molecular biology, you've got to start with the DNA. And what you have to do is you have to make a DNA template that represents the message, a cDNA. And we just need a short piece of cDNA from each of the genes, coded downstream of either a T7, a T3, or an SP6 promoter. Now the idea behind these promoters is that they come from bacteriophages, and they're actually DNA-dependent RNA polymerases. And the one we're going to use is T7. So it recognizes a particular footprint, and then the polymerase run copies of DNA into RNA from the 5' to the 3' end. And so what we have to do is we have to make a target DNA mRNA that has sites for the internal RT primer, 2 amplification primers, the forward and the reverse, and a TaqMan probe. So let's go back to that slide again. So we need to make a DNA that is going to be used to make this RNA here. Rightio. And then we have to make the synthetic mRNA DNA free, and so we use DNAs to get rid of the template. And then we quantify the RNA so we know how many micrograms per microliter we've got. And because we know the molecular mass, because we've got the sequence of the actual mRNA, we can work out the number of moles we've got, and thus the number of molecules. So we start with a plasmid called a PT7 gRNA. And this was originally designed to make guide RNAs for Cas9. And what we decided to do was actually to cut out the guide RNA bit and just use the T7 promoter to run off our RNAs. So what we did is the idea is to cut between the BSMB1 site and the BAM site and clone in your favorite gene. Okay, so it runs downstream of this T7 promoter. So this is what we did. This is Pkhd1 again, and then this is how we made the insert. We've got a forward primer for Pkhd1, so those will prime on a Pkhd1 cDNA or plasmid. Okay, we have a BSMB1 site here, a little extension here so that the BSMB1 can catch on to the PCR generated DNA, and then it will cut and give us this sticky here, TAGG. On the other end, we're going to use BSMB1 again, but this time it's going to create a BAM overhang, GATC. Okay, and this is the 5' overhang here for BAM-H1. And we're going to stick it into the plasmid. So we take the plasmid, the vector, and we cut with BSMB1 and BAM. So this generates these stickies here. And then we take the PCR product generated by this one here, and just slot it in. Okay, and we actually use a technique called Golden Gate. And there's actually a little talk on that. It shows you how to do it. So in fact, it's a one-tube reaction. You just mix your plasmid with BSMB1 and T4 ligase, and then you cycle between 16 degrees and 37 degrees 4 or 5 times, and everything that comes out from that reaction is perfect and will transform into E. coli. And you do have to sequence it, but 9 times out of 10 it's correct. And so this is how we made the PCR product. We use Q5 polymerase from NEB. It hardly ever makes mistakes. And we use either reverse transcribed RNA or plasmid template. And here you can see we've made a template that's about 550 base pairs long. And this PCR was very robust. It worked between 55 and 67 degrees, just from a reverse transcribed RNA. We then cloned that into our vector using BSMB1 and BAM. And then we cut the vector of BAM, and as you can see, we have a relatively large plasmid here. We've got one for Pkhd1, one for Pkhd2, one for Pkhd1, one for EPSIP, which is C21orf62, and this is a gene that I'm quite interested in, C16orf89. It might interact with EPSIP. I'm not sure though. So we cut with BAM, and so that linearizes it. So you've got your T7 promoter, your gene of interest, and then a BAM site. So the polymerase will go for the T7 through your gene of interest, making RNA, and it'll fall off. Okay. And then to get rid of any RNases, before we did the RNA step, we cleaned up with 0.1% SDS, 1 millimolar EDTA, and some proteinase K overnight, 1 microgram a mil. And then we cleaned it up. So how do we make the RNA? Well, the first thing we do is, once we've actually cloned our insert into the plasmid, we sequence the insert and ensure it's correct at the nucleotide level. And they always are with the Q5 polymerase. Then we cut with BAM-H1 in cut smart buffer, 37 degrees for 1 hour. Then treat with the proteinase K, as we stated in the last slide, overnight. And then we clean up with a PCR cleanup, and we use 1 microgram of BAM cut plasmid and use a standard RNA synthesis kit from NEB. So this has basically 2 tubes. One is a mix with your nucleotides, and one is your T7, and you basically follow the instructions and mix them together, and you give it 1 hour at 37 degrees. Then add DNase for 15 minutes at 37 degrees to get rid of the template. And that stops the reaction. So all you're left with in the tube is a high concentration of the RNA that you want. And then you do an RNA cleanup column. And this is what you get. We're using an RNA screen tape assay, tape station. And what you can see is we've got lovely RNA. It's about 500 base pairs along. And we can also quantify how much we've got. Okay, so in this case, for EPSIP, the C21 or F62, we've got about 0.3 micrograms per microliter. So quite concentrated RNA. What we like to do is we like to use the Pkhd1 fluorimeter to determine the concentration of RNA. And then we determine the molecular mass using a molecular weight calculator, this web page here. And then we determine the number of moles we have in a microliter by dividing the cubic mass by the known molecular weight of the actual RNA molecule, and then multiply by Avogadro's constant to give you the number of molecules per microliter. And usually we end up with about 10 to the 11 per microliter, okay? Now, once you've got your RNAs, you've then got to make your standard curve, and this is basically your standard curve that I generated for Pkhd1. But you're going to actually not just do one message, you're going to do 4 messages. So you've got a calibration curve for 4 messages at the same time. Okay, this is a bit complicated. All right, so here we develop a standard curve, and we're going to go from 1 copies a microliter to about 10 to the 10 copies per microliter. And so we need a carrier, and we're looking for another complex mammal, and I chose the dog because we won't be working with dogs. They're too cute. But we will be working with humans and mice. And so what we use is a Martin Darby canine kidney cell line, and we make RNA from that, and then dilute that RNA to 40 micrograms per mil in DPC water. So you've got about 12 mils of that. And then you dilute your standards from 10 to the 10 copies per microliter. So you take your roughly 10 to the 11 concentrated RNAs, okay, and then dilute them so that each of the RNAs is at 10 to the 10 copies per microliter in the same tube with the MDC carrier as the dilutant. Then you do 1 to 10 dilutions right down to 1 copies per microliter. And then you've got to have a blank as well. Okay, so a node template in the MDCK RNA control, 0 copies blank. And this is just to remind you of what we're doing. We've got 4 of these RNAs in there, okay? And then we're going to do a reverse transcriptase reaction with this internal primer, okay? So that's the next step. And this is the actual RNA sequence that we've got, and so this is that internal primer again. You see there are no T's, it's all U's because it's RNA. Okay, and so this internal primer will copy this RNA and make it into antisense DNA, and then cover this area as well, all right? And then you've got your forward primer and your reverse primer and your TaqMan probe. And this is labeled with a Yacamina yellow, which is like Vic, and it has a quencher, okay? And the molecular weight of this whole thing is 199.13 kilodaltons, okay, exactly. And so you use this to correct your micrograms per mil to the number of moles per mil or microliter, and then you multiply by Avogadro's number to give you the number per microliter. So now you've got to do a reverse transcriptase assay, and we use superscript 4 reverse transcriptase. We use about 10 microliters of RNA or standard, and if the RNA is from an animal or a human being, it must be DNase to remove genomic DNA, because some of these TaqMans haven't been made across intron-exon boundaries. They would pick up genomic DNA. So you also have to do an RT negative control to make sure that you're not picking up genomic. But if you've DNased them properly, you don't have any amplification from the genomic. The DNase is highly efficient. What we do is we add 1 microliter of internal primers at 2 micrograms per mil each in depth water. Okay, so that's a primer for the Pkhd1, the Pkhd2, the Pkhd1, and the C21orf62. Then you add 1 microliter of DNTPs, 1 microliter of water, anneal at 65 degrees for 5 minutes, and then chill it on ice. Now the second step is to add 4 microliters of the 5 times superscript 4 buffer, that 1 microliter of 0.1 molar DTT. Now this is important. We're going to have to dilute the sample because DTT will inhibit the TaqMan assay. You want 1 microliter of RNase inhibitor, okay, and it needs reducing power in order to be active, and that's why you have the DTT here. And then we use 0.5 microliters of the superscript 4 and 0.5 microliters of water. Well, we go canny on the superscript because it's expensive. We incubate at 55 degrees for 10 minutes, inactivate at 80 degrees for 10 minutes, and add 140 microliters of water and freeze at -80. So that converts your RNA to DNA. Now you need to do your TaqMan assay. To do this, you bring all your primers and TaqMan probes to 100 picomoles per microliter. You dilute your primers, unlabeled primers, at 1:200, and your TaqMan at 1:400 in prime time gene expression master mix. And you make enough for 10 microliters per well. So if you're just doing the standard, you're going to do everything in triplicate, and you'll have 10 wells. So that's 30 wells. 30 times 10 is 300. So you might make up 350 or 400 of the master mix. You put 10 microliters of the mix in each well, and then add 10 microliters of the diluted RT reaction per well. Treat everything the same, okay, so that you can come back to the standard curve without having to do any fancy calculations. Then we use an Applied Biosystems microamp EnduraPlate optical 96 well clear reaction plates for barcode, okay, and this is important. This number here is important because if you use the wrong plate, it won't run in the Quant Studio 5, okay. And then we cover it with a microamp optical adhesive film and then spin it in a centrifuge just to get everything down to the bottom. Then we run in the Quant Studio 5, annealing at 57 degrees for 40 cycles, and then collect the FAM, the VIC, the TAMRA, and the Sci5 channels. And so what you end up with for the standard curve is something like this, and the CT is about here, you know, at about 0.4. This is the logarithmic scale. So this is, the reactions are going exponential here, and then they're flattening out. So you want to catch them just before they begin to flatten out. And when you do, sorry, let me get to the bottom here. And when you do, you end up with a plot like this. And so what this is a mean CT. We've done everything in triplicate. And then we've got the log 10 number of molecules per microliter. So here you've got 10 to the 9 per microliter. And you're crossing at maybe about 15, a CT of 15. And here you've only got about 1,000 molecules per microliter, and you're crossing about 32, 35. Now, EPSIP, Pkhd2, and Pkhd1 are all behaving very, very similarly, but the Pkhd1 seems to be slightly more efficient than the others, perhaps because the primer's better and it primes more efficiently. But the point is that if you have unknowns and you have a CT, you can work out the number of molecules you've got per microliter for each of these genes and each of their mRNAs. So this should fill in some of the blanks that we have about cystogene expression. We know that the kidney and the pancreas and the liver all have Pkhd1, Pkhd2, Pkhd1 transcripts, but we're not sure how much they've got. And we don't know how they respond to injury or to hypoxia or to cystogen expression. And so an assay like this will be able to help us in the future. And so with this, I'll end the talk. And if you've got any questions, please just drop me a note and I'll try to explain. And if you like this talk, please give me a thumbs up, and thank you very much.